Hello students. So today let's discuss this very important topic of biotechnology application, and that is RNA interference. So it's a very interesting topic as well as important also. So if we go through this NCERT lines, so in this paragraph, in the first part of NCERT lines, it is talking about a problem, and the problem is the infection of tobacco plant by nematode. Then in the next few lines, it says that how this problem will be solved. So it says that RNA interference is the solution for this. And here they have given some basic introduction of RNA interference. Then in the last part, they have explained in detail the exact mechanism how nematodes are targeted. So in this case, we will see all these three headings. First, the problem, second, the basic introduction, and then how to target the nematodes. So most of the question in this topic will come from either of these three parts. Given in NCERT properly, there are few gaps which we will see it in detail. So let's start from one by one. So what is the first? The problem. The problem is of nematode infection. So as you can see here, there is nematode which is infecting. So first, nematode infect what organism? So first line, as it says in NCERT, nematode infect plant, animal and humans. Keep it in mind. But here what is the problem? Here this nematode is infecting which plant? It is infecting tobacco plant. Which part of the tobacco plant? Root of the tobacco plant. What is the name of this nematode? So name of this nematode is Melodygyne incognitia. So these simple point you remember. What happened due to this infection? So there is a decrease in the crop yield. As you can see, this is a diagram which is showing, this is showing the nematode. Now nematode, you know, it is in animal kingdom, you might have studied it, that they are the animals. This is the tobacco plant, and this is how the root infection look like. As you can see, this is the infected root and this one is the normal root. This diagram is given in NCERT. So basic point, once again, if I highlight here, you have to just remember nematode infect plant, animal as well as human. Nematode, which is Melodygyne incognitia, it infect tobacco plant. Which part of tobacco plant? Root. And what it does? It reduce the yield of tobacco. Now, so if we have understood this problem, then what is the solution of this problem? So the solution of this problem is a novel strategy, and that novel strategy is called RNA interference. Then now the question comes, what is RNA interference? So see, RNA exists naturally in naturally in nature, right? It exists naturally in nature. So how does it exist? First, where it exists? In eukaryotes. How? As a method of cellular defense. So remember, it is not found in prokaryote, found in eukaryote, and for which purpose? For cellular defense. Then comes, what does this RNA interference target? So this RNA interference target specific mRNA. Now, what do you mean by specific mRNA? So for that, let's first understand this. There is this double-stranded DNA, right? Now, this DNA, after transcription, let's say it form a mRNA. We have already studied that transcription form mRNA, and this mRNA will form protein, right? Let's say this is the protein. So this is the simple central dogma also we have studied, that there is transcription and then there is a translation. Now let's consider here. Let us say this protein, let's name it X. This X protein is very important. Without that, the organism will not survive. Now, due to some reason, if we stop the formation of this protein, then will the organism survive? No. If we inhibit this protein, then the organism die. Now, so to inhibit this protein, what we will target? We will target the mRNA. So, in this RNA interference, our purpose is to inhibit this protein. And for inhibiting this protein, what we will target? We will target mRNA. So that's why RNA interference is silencing of a specific mRNA. Now, which mRNA? Which mRNA? Whose protein we want to inhibit? Whose protein we want to inhibit? Now, question come, which protein we want to inhibit? So we will choose a protein which is very important for the cell to survive. That protein we will target. So now we understood the target. Now if I ask you which process it will affect, so it will affect translation because mRNA is already formed. So transcription is already done. So we are targeting translation. So which process we prevent? We prevent translation. So what is our target? Our target is mRNA and we prevent translation. That is why we also call this RNA interference as a post, note it down, post transcriptional event. Post transcriptional event, because it do not affect transcription, it affect translation. Then question comes, how does this mRNA is targeted? So this mRNA is targeted by a specific double-stranded RNA. So till I will explain how this double-stranded RNA targets it, but now from here in this case, how many points you will remember? So you have to simply remember few things. I will make it very clear. That first thing you will remember is that, as it is mentioned here, I will highlight that it is found in eukaryotes, right, as a method of cellular defense. What it targets? So it targets the mRNA. So which process it targets? So it targets the translation. How it target? By forming a double-stranded RNA. This double-stranded RNA will act on mRNA, right? And it cause silencing of mRNA, right? It will cause silencing of mRNA because it takes place after transcription. So it is called post-transcriptional event. So these all points you have to remember simply. Now the question comes that how this double-stranded RNA is formed and how it will target mRNA. So let's see that one by one. So now if we see how this double-stranded RNA is formed. So now double-stranded RNA can be formed naturally, as I say here. So it may be formed naturally. Now how it is formed naturally? There are certain viruses, right? And which virus? RNA genome. They will insert the RNA inside the eukaryotic cell, right? They will insert the RNA inside the eukaryotic cell, and these RNA will inhibit the mRNA. So the source of this double-stranded RNA, it is virus in some case, and in some cases it is transposon. Transposon is also called as mobile genetic element, or it is sometimes also called as jumping genes. Now the question comes, what is this jumping gene or mobile genetic element? Although not given properly in NCERT, but just understand. Let's say this is a double-stranded DNA, right? Now in this, if I assume that this is a small part of it and this is a segment of DNA and this is called transposon. Now this transposon have this character that it will change its position from one place to another place, and that is why it is called jumping genes. And they will interfere, means they will form the RNA which will take part in RNA interference. So now from here what we understand, that there are some more, but in NCERT because they have given two, we will limit ourselves to two. So first source virus, second source transposon. Which virus? Virus with RNA genome. Now apart from this, this is natural, but now we also develop transgenic plant. We develop transgenic plant, and these transgenic plant will form double-stranded RNA to target specific mRNA. To target specific mRNA. So again, here in this part, if I summarize, so this double-stranded RNA is formed from either virus or from transposon, and if we are doing it, then we will develop a transgenic plant which will form double-stranded RNA which will target a specific mRNA. And this is exactly what we are going to kill the nematode. So now we are seeing how we are targeting the nematode. So now in this step, this is the last part of NCERT where we are seeing how we will kill the nematode. So our main purpose is to kill the nematode. What is the name of the nematode? In the beginning we have seen it was Melodygyne incognitia. So our aim is to kill the nematode. Now for that, what we will need to do? So we will select a protein. We will select a protein without which nematode can't survive, right? If I inhibit the protein formation, then nematode will die. Now, so this protein will be formed by a mRNA, isn't it? So actually to inhibit the protein, we will target what? So we will target mRNA. We have already seen we will target mRNA through RNA interference. So this is basic, we have already seen. So main point is in nematode we have to target a protein, and for targeting the protein we need a mRNA. Now, so how to target this mRNA? We have already seen we will target the mRNA by forming a double-stranded RNA which will target the mRNA. Now question come, how we get double-stranded RNA? So as I have said, we will create a transgenic plant, right? So why we are creating a transgenic plant? We are creating a transgenic plant to form a double-stranded RNA which will target the mRNA. Now how to develop a transgenic plant? So understand here, in the previous example of BT cotton also you might have already understood that there is a bacterium called agrobacterium, and this agrobacterium has a specific plasmid, and that plasmid is called TI plasmid. Now this TI plasmid I will use to insert a nematode specific gene, right? This is the nematode specific gene which I will insert it into the agrobacterium. Now what is the meaning of nematode specific gene? See, nematode specific gene means now this gene will form the double stranded RNA which will target the mRNA, right? Which will target the mRNA. Understood? So this nematode specific gene will form a double stranded RNA. Now question comes that how this nematode specific gene will enter into the plant. So you know already by now that this agrobacterium transfer the desired gene into plant cell, transfer the desired gene into plant cell. Transfer the desired gene into plant cell. So my nematode specific gene now entered into the plant cell. Now inside that, let us say this is a nematode specific gene. Nematode specific gene means it will be a DNA. So this is my nematode specific gene, that is a DNA. And where it is present? It is present in the plant cell, right? It is present in the plant cell. Now in plant cell, actually this DNA will form two types of RNA. It will form a sense RNA and it will also form a antisense RNA, right? So I will write it here that it form a sense RNA and it will form antisense RNA. Now by now you must be clear that a sense and antisense RNA will always be complementary to each other, and this sense and antisense, they will come together and they will form a double stranded RNA, right? The sense and antisense, they will come together and now they will form a double stranded RNA. Understood? Now here see, You will think that, sir, how can DNA form a double-stranded RNA? Usually DNA form a single-stranded RNA. See, DNA here is forming single-stranded RNA only, but it is forming a sense and antisense which are complementary to each other, so we get the double-stranded RNA. So till here every point is clear. How we have created double-stranded RNA in a plant cell? So once again, in a very simplified way, let me explain this. So basic purpose of developing a transgenic basic purpose was that you need a plant cell which form a double-stranded RNA, and this double-stranded RNA should target mRNA. mRNA of nematode, not mRNA of a plant cell. But the double-stranded RNA will be formed in the plant cell. Now question is how this double-stranded RNA is formed. So it is formed from a nematode specific gene. How we will introduce this nematode specific gene into the plant cell? With the help of agrobacterium. Why we choose agrobacterium? Because agrobacterium has Ti plasmid. Now, once this nematode specific gene is transferred into the plant cell, this DNA will produce a sense RNA and a antisense RNA. Because they both are complementary to each other, they form the double-stranded RNA. Now, if I ask you this question, double-stranded RNA is formed in, and if I say it is formed in agrobacterium, or it is formed in plant cell, or it is formed in nematode. So where you will say it is formed in plant cell. Further, one more question if I ask you that the transgenic plant form, let's say this is the question, transgenic plant form, and I give you the option, it form sense RNA, it form antisense RNA, third option it form double-stranded RNA, and fourth option all of the above. What you will say? So you will say all of the above. Why? Because of course sense RNA is also formed, antisense is also formed. They both together is forming double-stranded RNA. Now, sometimes just to confuse you, they will give you sense DNA. Now, is sense DNA formed? No. No sense DNA is formed. No antisense DNA is formed. So be very aware. The same question is asked, can be asked by giving option as sense RNA or sense DNA. Now, if sense DNA is given, it is wrong. Antisense DNA is given, it is wrong. So in this way we get the double-stranded RNA. Now next part is what? Now this double-stranded RNA will go and target the, so it will go and target the mRNA, but where? In the nematode, right? Now the question comes, how this double-stranded RNA which is formed in plant cell will enter into the nematode? So that's a good question to ask, right? Now the big question is, how does this double-stranded RNA enter into the nematode? So understand, nematode, you know, we have already seen nematode attack the tobacco plant. Which part it attack? It attack the root. So it feed on the transgenic plant, and when it feed on the transgenic plant, double-stranded RNA produced is ingested by the nematode. So let's say if this is the nematode and it attacks the root, so it will ingest the double-stranded RNA. And when it ingests the double-stranded RNA through the gut, it will enter into the cell. So very simple question, how does this double-stranded RNA enter into the nematode? So simply by ingestion, and it through the gut it will enter into the cell. So now the double-stranded RNA is present inside the cell. Now here there is one point where some of you get confused, which NCERT is not clarifying very much. NCERT simply says that this double-stranded RNA goes and it silences the mRNA. It silences the mRNA. Now many of you think that, sir, mRNA is single-stranded. How a double-stranded RNA can silence a single-stranded mRNA? And your doubt is correct. NCERT has skipped few steps, right? So which is I am clarifying here. Now see, this double-stranded RNA is present inside the nematode. Now this double-stranded RNA will not directly go and attack the mRNA. This double-stranded RNA will be first affected by the dicer protein. See, Let's see it this way. So let's say, for example, we have this double-stranded RNA. Now, this double-stranded RNA will be acted upon by a protein, and this protein is called dicer protein. This dicer protein will go and digest and break down this double-stranded RNA into large number of small fragments of RNA, and that is called siRNA. It is also called as small interfering RNA. Small interfering RNA. Now, this small interfering RNA will go and bind to the mRNA. Now, you can recall there in the beginning we have choose one mRNA as a target, right? Which was forming a very important protein, isn't it? It was forming a very important protein. Now, what happened? These small interfering RNA will come and join with the mRNA. It will come and join with the mRNA, and it will form several small double-stranded part. Now, due to making of this regular double-stranded part, what will happen? Translation will be interrupted, and when translation is interrupted, the protein formation will be blocked. So if there is no protein, then what will happen to the nematode? Now the nematode will die. Nematode will die. So in this way we have killed the nematode. What was our purpose? Our purpose was to kill the nematode, and this is how we have actually killed the nematode. If we see it step by step, so what we have done first, so first, if I am writing it one by one, so first what we have did, so we have transferred nematode specific gene into plant cell, Now it is very clear how it was transferred. So it was transferred using agrobacterium. And why agrobacterium? Because agrobacterium has Ti plasmid. So this is the first step. Now inside the plant cell, what is formed? Inside the plant cell, the DNA forms. DNA means the gene, right? DNA form sense RNA, and it form antisense RNA. And because of that, what is formed? Double-stranded RNA is formed. Now, third step is this double-stranded RNA, it is ingested by whom? It is ingested by nematode. Now the double-stranded RNA is inside the nematode. What will happen now? This double-stranded RNA inside the nematode, this double-stranded RNA is broken into small interfering RNA by which protein? By dicer protein. And ultimately, lastly, this siRNA binds to the mRNA. It binds to mRNA and it causes silencing of mRNA. It will cause silencing. So what happened? No protein formed. And if there is no protein, then the nematode dies. So this is how we will do the RNA interference. So if you just keep these steps in mind and all the points in mind, then you can easily solve all the questions from this topic. So in the description here, I have given some question related to RNA interference, which you should solve. If there is any doubt, you can comment and you can ask.